All right, guys, so this is video 2 of the scientific method lecture. So we're going to go ahead and pick up where we left off. So in the last video, we talked about collecting data. All right, we can collect qualitative data, which is using our senses to make observations to collect data, red apples, red tomatoes, and whatnot. And then quantitative, how many there are, using actual numbers. Now notice here it says SI units must be included in all measurements. Well, hopefully you know by now that if you do not have units on the end of numbers, then the numbers are meaningless. For example, if you're measuring the mass of the tomatoes and you do not put grams on the end of the number, then everybody's going to think it's just a number. So what is that science is talking about? So you have to have SI units. And SI, not to be confused with the English system, so not pounds, like miles, things like that. You know, in the SI system, we talk about grams, liters, meters, and that kind of thing. All right, so your data must be kept orderly in a table or chart. So whenever I ask you to collect data, please remember that the data needs to be in a table or in a chart, or it's not going to be organized. It's not going to look neat. Because ultimately the purpose of this is for someone else to look at and verify your science to make sure that it is accurate so that it can be accepted. Modify the procedure if needed. All right. Step 6, you're going to analyze data, confirm the results by retesting if possible. Trials, the number of times you repeat the experiment. So if I'm doing an experiment, I only do one trial. That's just one set of data. But what does that mean if I recreate those conditions and I get the same results? And then I do it again and get the same results. So you have to have several trials. The more trials you can do, the more reliable your results. If I do something and get this result and do the same thing again and get a different result and then a different result, then there's something else going on, you know, other than what I hypothesized to be happening. So then here's an important thing that I need to point out to you. So if you're using DocHub or a PDF editor to make notes, highlight this, convert results to a graph that is appropriate for the experiment. Why? So that everybody else can see it. Graphs make things more accessible, easier to read. You might watch the news by accident or on purpose, or you might read something, say about the coronavirus, and then you have a certain number of cases. You know, you see the graph goes up, but then something happens in the graph, shows a downward progression, so the slope goes down. Or you've probably heard by now, flattening the curve. So you can really see the flattening of the curve if you have a graph. It's kind of hard to see that without a graph. So graphs are highly important and must be used when you analyze your data. And then lastly, use both descriptive and inferential statistics to help make conclusions. So that's something that we're going to talk more in-depthly about in AP Biology. So more to come on that, but not so much in Earth and Space Science. In AP Biology, it's a reoccurring thing that you'll see on tests and quizzes in the future. So statistics, math, very important in all sciences. All right, next, form a conclusion. The written results of the experiment include a statement if the hypothesis was supported or refuted, which is kind of the whole purpose, right, is to solve or collect evidence to support your hypothesis. Way back in the 1600s, Galileo Galilei was number one in that area. He determined that if you have a question and you have an answer to that question, your answer is meaningless unless you have evidence, objectively obtained evidence. If you don't know what the word objectively means, Google it, look it up. It's really important. Kind of the opposite of subjectively. Basically, it just means thinking with evidence, thinking unbiasedly. Very important. All right, and then here we have make recommendations for further study and possible improvements to the experiment. And then, what is a science experiment if you do not communicate results? I'm going to be doing experiments up here, and I'm going to be asking you to communicate your results. Your results are meaningless. I mean, if you do not communicate them to other people to have them critique, analyze your result, and especially if you're a scientist, that's your job. That's how you make your money. You communicate your results. Very important step there, collecting your money. All right, so the next thing that we talk about is graphing. Now, if you remember, graphing relates to which step? Right here, C. Convert results to a graph that is appropriate for the experiment and analyzing your data. So graphs and charts communicate information visually. They can show patterns, help scientists identify correlations, and get the point of the experiment across quickly. The independent variable is usually plotted on the X, dependent is usually plotted on the Y. So you have to remember, what does independent and dependent mean? Now, this is where they go on a graph. The mnemonic dry mix for dependent responding Y-axis and manipulated independent X-axis can help you remember this pattern. Dry mix. Remember it. You're going to label both axes, the independent variable on the X and dependent on the Y. Again, include units on both axes. Why is that important? Oh yeah, we said that a number without a unit is just a number. It doesn't mean anything. Enclose the units in parentheses. All right? So if I'm measuring distances per something else, then I'm going to have whatever I'm measuring the distance in. SI unit would be meters, kilometers, in parentheses. Provide a descriptive title. Use the pattern, the effect of the independent variable on the dependent variable, the effect of Miracle-Gro on the growth of plants, the effect of wearing a mask on the transmission of the coronavirus or microorganisms in general. So on. If the instruction is to plot rather than graph the data points, no line needs to be drawn. So you might do a dot graph. All right, moving on. So here's where it talks about using a line of best fit. So at some point we will do that, and we will talk more about it once we get there. And then you will decide, you'll have to decide based on your data what type of graph you should use. So should you use a line graph, bar graph, scatter plot? By this point, you guys have graphed a lot, but you have to understand that some data doesn't need to be in a line graph. It wouldn't make sense, because line graphs are used for looking at relationship between two continuous types of data. Typically, both the independent and dependent variables are numerical. And what you use for a line graph may not work for a bar graph or a scatter plot. Scatter plots are used for examining relationships between two types of data. These are very similar to line graphs, just without the line. Line graphs provide an excellent way to map independent and dependent variables that are both quantitative. When both variables are quantitative, the line segment that connects 2 points on the graph expresses the slope, which can be interpreted visually related to blah, blah, blah. All right, moving on. So that's graphs. And that is the scientific method. It is very important. And I know that these videos were rather long and there's a lot of information in it, but I'm stressing to you that the number one thing in science class to learn is the scientific method. And you think about, all the things that are debated in the world, that are talked about. For example, the CDC. Why does the CDC change their rules, their procedures for the coronavirus? Well, as scientists who work for the CDC collect more information, they have to change those regulations, those distances, to wear a mask or not to wear a mask, So as you collect data, science informs you, you make new conclusions, therefore you make new rules. So that is why what the CDC says changes. That's why science, when I was your age, has changed significantly from when I was there, and then in 10 years it'll change a lot for you. So some of the things that you learn in high school may be different. If you remember in the 9th grade when you learned about Punnett squares, I learned about Punnett squares also in the 9th grade, but now they are to an extent obsolete because we've learned genetics is way more complicated than that. So that's it for the scientific method. Now the next step, after you watch the 2 videos that I have here for the lecture video, I want you to go in and complete this scientific method assignment that is due Friday. So if we click into it real quick, I placed instructions. After watching the video, How Well Do Masks Works, use the Google Forms to explain how scientists in the video use the scientific method. Make sure to go through each step of the scientific method and relate it to the video, starting with the first step of the scientific method, which is ask questions. So if we pull this up, what I'm asking you to do is design an experiment to answer the question, how well do masks work? So you're going to state that question, make some observations. Go back to the video where I talked about making observations. You're going to formulate a hypothesis, design an experiment, collect data, analyze data, form conclusion. So you're not actually going to be required to do this experiment. It's more of a thought process or thought experiment. Tell me, if you were going to solve that problem as a scientist, how would you collect objective evidence? All right. So let me know if you have questions on that. Hopefully it's not all too confusing, but basically go through and complete each step of the scientific method. All right. I hope this was helpful.